Hello students, welcome to EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today we are going to talk on module Lipid Oxidation from the paper Lipid Metabolism. The objectives of the paper includes to understand how the oxidation of lipids takes place and what are the steps that are involved in the fatty acid oxidation. Before understanding, let us know the role of hormones in the mobilization and transport of fats. How after digestion and absorption process the lipids molecule mobilizes and transported in the tissues or the cells. The triacyl glycerol stored in the adipose tissues are mobilized. That the idea is to brought it out of the storage and transport it to the tissues including the skeletal muscles, the heart and the renal cortex, in which the fatty acids are oxidized for energy production. Epinephrine and glucagon are the two hormones that are secreted in response to the low blood glucose levels. These two hormones activate the enzyme adenylyl cyclase in the adipocyte plasma membrane, which produces the intracellular second messenger cyclic AMP or the cyclic adenosine monophosphate. This cyclic AMP dependent protein kinase A then phosphorylates perilipin A, which allows the hormone sensitive lipases in the cytosol to move to the lipid droplet surface, where it can begin hydrolyzing the triacyl glycerol to free fatty acids and glycerol. The protein kinase A also phosphorylates hormone sensitive lipase, thereby increasing the activity of it 2 to 3 times. But more than 50-fold increase in fat mobilization triggered by epinephrine is primarily due to the perilipin phosphorylation. Cells with defective perilipin genes have almost no response to increase the cyclic AMP concentration. Their hormone-sensitive lipase does not associate with the lipid droplets. In this concept map, you can see the network of glycerol, triacyl glycerol, fatty acid, carbon dioxide and water, and how these are broken down to form various products. See the triacyl glycerol and the fat molecule simply broken down to glycerol and fatty acid. Then further, fatty acid, the fate of fatty acid breakdown is the ultimate formation of the carbon dioxide and water. However, glycerol has been utilized for the glycogen production. The fatty acid is either converted to unsaturated fatty acid or saturated fatty acid, then saturated fatty acid, even number carbon, and odd number carbon fatty acid, then unsaturated fatty acid to monounsaturated fatty acid and polyunsaturated fatty acids, and finally even number carbon saturated fatty acid broken down to shorter chain and a bigger chain, whereas the odd number carbon is broken down to acetyl-CoA, palmityl-CoA and succinyl-CoA. Then these succinyl-CoA via the Krebs cycle or the TCA cycle generates energy. However, the bigger chain utilizes the carnitine shuttle system to undergo beta oxidation. The odd number double bonds are handled by the isomerases and the even number carbon ones handled by the reductases and isomerases both. In this picture, we can see how the triacylglycerol, through the process of esterification and lipolysis, broken down to fatty acids, then through lipogenesis as well as beta oxidation, gets converted to acetyl-CoA, which further lipogenesis form fatty acid, and these acetyl-CoA is involved in cholesterologenesis as well as ketogenesis to form cholesterol and ketone bodies respectively. The cholesterol through steroidogenesis results in the formation of the steroids, and the acetyl-CoA enters through citric acid cycle to form carbon dioxides. A very famous concept for the beta oxidation of fatty acids has been given by Knoop. Actually, the earliest theory on the fatty acid oxidation was the beta oxidation hypothesis, and that was given by Franz Knoop in 1904, almost 111 years back. He hypothesized that the fatty acids chain is degraded in a stepwise fashion by the repeated removal of the two carbon atoms due to the oxidation at the beta carbon atom. For example, an 18 carbon chain stearic acid would be the first to be oxidized to 16 carbon palmitic acid, and after that to 14 carbon, followed by 12 carbon, 8 carbon, 6 carbon, 4 carbon, 2 carbon, and ultimately to carbon dioxide and water. Many researchers have added knowledge to the mechanism of the fatty acid oxidation, but the fundamental concept given by Knoop is still valid. The beta oxidation of long chain fatty acids is vital to the prerequisite of energy for the organism and is of fastidious substance for cardiac and skeletal muscle. However, other organs, principally the liver, but also the kidney, small intestine, white adipose tissues, can utilize the product of beta oxidation for the formation of ketone bodies, which can in turn be utilized for the energy by other tissues. The relationship of lipids or the fat oxidation with the utilization of carbohydrates as a source of energy is complex and depends upon tissues, nutritional state, exercise, development, and a number of other factors such as infections and other pathological states. The Knoop's experiments, the mystery of the product of odd and even chain fatty acids. As elucidated by Knoop, the mechanism of fatty acid oxidation was stated to be the oxidation at the beta carbon of the fatty acids. Knoops performed several experiments in dogs by feeding them with the odd number carbon fatty acids as well as with the even number of carbon fatty acids. The landmark experiments of Knoop's results in the Knoop's concept of fatty acid metabolism. His experimental details: by feeding dog with odd number carbon fatty acid results in the formation of phenylpropanoic acid as well as phenylvaleric acid. He isolated hippuric acid from the urine of the dogs fed with the odd number carbon fatty acid, and this hippuric acid is the conjugate of glycine and benzoic acid. In another experiments, by feeding dogs with the even number of carbon fatty acid, he identified the phenylbutyric acid, and he isolated phenylaceturic acid from the urine of the dogs fed with the even number carbon chain fatty acid. From his experiments in dogs, Knoop proposed that the oxidation occurs at third carbon, which is the beta carbon, resulting into beta keto acids which yield fatty acids shortened by 2 carbon atoms. The Knoop experiments were confirmed by Dakin, who performed similar investigation. Emden and co-workers established that unsubstituted fatty acids are metabolized by beta oxidation to ketone bodies in perfused livers. Again, experiments in cell-free system confirmed and elucidated that the whole mechanism of beta oxidation through the experiments of Munoz, Leleur and Leninger. One important aspect through the work of Leninger unveiled the energy spark in the form of ATP, which is required in beta oxidation. The major simplified steps of beta oxidation of fatty acids. In the beta oxidation of fatty acids, the first step is the activation of the fatty acids by the formation of the coenzyme A derivative, which is ATP mediated. Next step of the beta oxidation of fatty acid is the desaturation between the alpha and beta carbon atoms. The active fatty acids are then oxidized by beta oxidation, and finally the formation of 1 mole of acyl-CoA and a fatty acid shorter by 2 carbon atoms, and because of a thiolytic split with coenzyme A already activated for the repetition of the oxidative cycle. The first reaction of the beta oxidation, which is called as the activation of the free fatty acid. Here we can see in this reaction that the free fatty acid along with coenzyme A and adenosine triphosphate forms acyl-CoA or the acyl coenzyme A, together with the formation of adenosine monophosphate and inorganic phosphate is liberated. This fatty acid from carbon 4 to 18 combined with coenzyme A, the reaction was catalyzed by thiokinase, and magnesium ions are essential to carry out the reaction. And in this way, in first reaction, active fatty acid is formed. The activated fatty acids undergoes beta oxidation in the mitochondrial matrix. After the fatty acids are activated on the outer mitochondrial membrane, the next problem faced by them is the transport from the outer mitochondrial membrane to the matrix. And this is done through the help of carnitine, which is also 3-hydroxy 4-trimethyl ammonium butyrate. This carnitine is a zwitterionic alcohol by the mode of conjugation reaction catalyzed by carnitine acyl transferase 1. This enzyme carnitine acyl transferase 1 is also known as carnitine palmitoyl transferase 1. This enzyme is bound to the outer membrane of the mitochondria, and it catalyzes the reactions in which the acyl carnitine is formed through the transfer of acyl group from the sulfur atom of CoA to the hydroxyl group of carnitine. This picture depicts how the fatty acid enters into the different compartments of the mitochondria. If we look at the reaction, we can see that carnitine along with acyl-CoA, the activated form of fatty acid, gets converted to acyl-carnitine and coenzyme A is released, and this action has been carried out by carnitine acyl transferase or carnitine palmitoyl transferase. At the outer membrane, the fatty acid gets converted into the active fatty acid, and in the intermembrane space of mitochondria, it gets converted to mannonyl-CoA, and along with carnitine it forms acyl-carnitine, and this acyl-carnitine through the outer face of the intermembrane space and the inner face of the matrix enters the matrix of the mitochondria, where the beta oxidation takes place. The second reaction of the beta oxidation, that is removal of two hydrogen atoms that require FAD catalyzed by acyl-CoA dehydrogenase. So now the activated fatty acid, which is in form of acyl-CoA, along with FAD gets converted to trans-enoyl-CoA and FADH2. This reaction is catalyzed by acyl dehydrogenase, where the flavoprotein oxidized form gets converted to a reduced flavoprotein form and alpha beta unsaturated fatty acid formation takes place. The third reaction of the fatty acid oxidation, the saturation of the double bond through water. The trans-enoyl-CoA or the alpha beta unsaturated fatty acid, which is formed in the second reaction of the fatty acid oxidation, acted upon by enoyl hydrase or enoyl hydratase, also called as crotonase or 3-hydroxyl acyl-CoA hydrolase, to form beta hydroxy fatty acid, also known as 1,3-hydroxyacyl-CoA. Reaction number 4 of the fatty acid oxidation, which is the dehydrogenation step involving the use of NAD+. 1,3-hydroxyacyl-CoA or the beta hydroxy fatty acid, which is formed in the third reaction of the fatty acid oxidation, together with NAD+, gets converted to 3-ketoacyl-CoA along with NADH plus H proton ion. The so formed beta keto fatty acid results due to the catalytic action of beta hydroxyacyl-CoA dehydrogenase. Reaction number 5 of the fatty acid oxidation, which is the splitting of ketoacyl-CoA to 2 carbon shorter chain length than the original acyl-CoA. The 3-ketoacyl-CoA, also known as beta keto fatty acid, along with coenzyme A, gets transformed into acetyl-CoA and acyl-CoA. The so formed acyl-CoA is shortened by 2 carbon, whereas acetyl-CoA again reutilizes in the metabolic pool. The reaction is catalyzed by beta-ketothiolase, also called as thiolase. It shortens the fatty acid by 2 carbon, and ultimately this active fatty acid again enters the beta oxidation to be further oxidized. Is the summarized view of the 5 reactions which are going on in the fatty acid oxidation process. How the fatty acyl-CoA gets converted to trans-enoyl-CoA through the action of acyl-CoA dehydrogenase. Oxidized flavoprotein gets reduced, and then this trans-enoyl-CoA, by the catalytic action of enoyl-CoA hydratase, which utilizes a molecule of water to get converted into 3-hydroxyacyl-CoA, which further acted upon by hydroxyacyl-CoA dehydrogenase that converts NAD plus to NADH and a proton and forms beta-ketoacyl-CoA. Finally, this beta-ketoacyl-CoA, under the catalytic action of beta-ketothiolase, utilizes a coenzyme A to form the fatty acyl-CoA as well as acetyl-CoA. This fatty acyl-CoA recycled again to the spirals of the beta oxidation, whereas acetyl-CoA is utilized in the metabolic pool. So students, in this lecture we have learned about how the oxidation of lipids or the fats takes place. We have also come across the Knoop's concept of beta oxidation. Though the theory has been conceptualized 100 years back, even more than 100 years back, it is still a valid theory for the oxidation of lipids. We have also learned about the implications of the reactions involved in the beta oxidation, as well as the role of carnitine acyltransferase in the fatty acid oxidation. Thank you.